Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the biology topics. This is plants and food. We begin by looking at an experiment for testing a leaf for starch. In this experiment, you will pluck the leaf from a plant. You then dip this leaf in boiling water. This is to remove the waxy cuticle. After the cuticle is removed, then we place the leaf into the ethanol, which is boiling. Initially, we're going to boil the water, but we do not boil ethanol directly. We'll place a test tube containing ethanol into the boiled water. Since water has a boiling point of 100 degrees and ethanol has 78, by the time the water is almost boiling, the temperature of ethanol can be raised to 78, which is sufficient for it to boil, and so the leaf can be placed in ethanol, which will be boiling. This is going to allow the ethanol to penetrate the leaf in order to remove all the chlorophyll, so the leaf will be decolorized. Removing chlorophyll enables us to see the color contrast after addition of iodine. In the next step, we're going to use cold water to wash off the ethanol as well as cool the leaf. And after that, we'll add iodine solution. Here we're placing the leaf onto a petri dish and then we add iodine. So the color is going to change. If there is starch, we'll see a blue-black color observed. And then if there is no starch, it's going to remain yellow-brown as the color of iodine. This is a negative test and that is going to be the positive test. Next, we'll look at photosynthesis. Photosynthesis is a process by which green plants convert sunlight energy into chemical energy. To do this, we will need carbon dioxide and water. In the presence of sunlight, this is going to be trapped by chlorophyll in order to convert these two into glucose as well as oxygen. For this to occur, here are the conditions. We need sunlight, we need CO2, and we need chlorophyll like I've talked about here. Next, we're testing the conditions necessary for photosynthesis to occur, and the first one is looking at chlorophyll. To do this, we will use a variegated plant. The variegated plant has variegated leaves. These have parts with chlorophyll and other parts without. For example, we can see here the green parts have chlorophyll and the other parts do not have any chlorophyll. We're going to expose this plant to sunlight, maybe about 24 hours later. I'm not talking about 24 hours worthy of sunlight, but allow it to photosynthesize for about 24 hours. And then you test for presence of starch in a leaf that is going to be plucked off from this plant. Since only the green parts contain chlorophyll, we expect them to turn blue-black. We're going to use boiling water to de-wax, and then we'll use boiling ethanol to remove the chlorophyll. And after that, we're going to add iodine. Then we'll see the color changes. The green areas are going to turn blue-black due to presence of starch, and the other areas are going to remain yellow-brown or yellow due to lack of starch, and this color is going to be due to the color of iodine, the yellow-brown color. So it will mean in these areas that we saw turn blue-black, there was photosynthesis that took place, and therefore chlorophyll is necessary for photosynthesis to occur. The next condition is carbon dioxide, studying if carbon dioxide is really necessary for photosynthesis to occur. Here we'll begin by destarching two plants overnight. We will put them in the dark so that they lose all the starch from their leaves. And then the plants should be of the same species and age in order for the experimental results to be consistent. These are going to be placed in bell jars, like we see here. There is a bell jar here and another bell jar here. And since here we are testing for carbon dioxide, we're going to expose them to sunlight. That's why the bell jar is not covered with aluminum foil. But in one experiment, we're going to put soda lime. The soda lime is going to remove carbon dioxide. In the other, there will be no soda lime, so CO2 is going to be present here. This plant is the control. It's going to have sunlight, it's going to have carbon dioxide, it's going to have water, it will have chlorophyll, and that means photosynthesis is going to occur here. Well, here photosynthesis will not occur because carbon dioxide has been removed, which is necessary for photosynthesis to occur. At the end of the 24 hours, we're going to pluck a leaf from each of the bell jars, and then we're going to test it for presence of starch. We expect this one here to have no blue-black color observed, or it's going to remain yellow color, which is the color of iodine, while this one here is going to carry out photosynthesis, and therefore we'll see a blue-black color observed in the leaf of that plant. Next, we study if sunlight is necessary for photosynthesis to occur. Again, remember the conditions we talked about are presence of chlorophyll, presence of sunlight, presence of carbon dioxide, and so on. So we've tested carbon dioxide, we've tested chlorophyll, and now we're going to test sunlight or light. To do this, we're going to get two potted plants. They have to be the same species and the same age. We will destarch them overnight. Here you could say 24 hours or just overnight in a dark room in order to lose all the starch from the leaves. And during the experiment, both plants are going to be kept in the dark. This is not going to be exposed to sunlight or light, but this one here is going to be exposed to light from one source. Like here, this is a light source, so that we can control the amount of light that this plant is exposed to. So in this experiment, the light source is going to be placed at the same distance from the experimental setup. And at the end of the 24 hours, we're going to pick a leaf from each of the bell jars, and we're going to test that leaf for presence of starch. We will expect starch to be present in here when we add iodine. This is the control experiment, and this is the test experiment. Here there is no light, and therefore photosynthesis is not going to occur, and that will show us that light is required for photosynthesis to occur. Next, we go to the structure of the leaf. This is useful for photosynthesis, and it has adaptations that enable it to carry out photosynthesis efficiently. It has a large surface area to absorb more sunlight, and we see many chloroplasts containing chlorophyll, and this is to trap sunlight in order for photosynthesis to occur. Due to the stroma, carbon dioxide is going to come in efficiently, and the products of photosynthesis are going to leave efficiently through the phloem. These are the tubes that carry away the products of photosynthesis. Now, the components we're going to talk about first is the epidermis. The leaf contains the upper as well as the lower epidermis. The epidermis contains a waxy cuticle for reducing water loss through evaporation, and it acts as a barrier against disease-causing microorganisms. Next, we go to the guard cells. These are mainly found on the lower epidermis, and they change their shape in order for the stomata to open and close. The stomata allow for exchange of carbon dioxide and oxygen, as well as water vapor loss. Next, we go to the palisade cells. These ones here, we can see they're aligned in a way closer to the surface of the upper epidermis in order to maximize absorption of sunlight. They are made up of narrow and long cells that contain numerous chloroplasts, and that means a lot of chlorophyll is contained and a lot of sunlight is going to be trapped. Then we go to the cells beneath them. These are the spongy cells. They are loosely packed with air spaces between them. The cells contain chloroplasts and can photosynthesize, but the chloroplasts contained are less than those found in the palisade cells, so less photosynthesis takes place in the spongy cells in comparison to the palisade cells. They form the main gas exchange surface in the leaf. Carbon dioxide is absorbed here and oxygen is going to be released. And then we go to the xylem and phloem vessels. The xylem are vessels that transport water and mineral ions from the soil through the roots to the stems and finally to the leaves in the transpiration stream. And the phloem, these transport products of photosynthesis away from the leaves towards other parts of the plant for use or for storage. Next, we go to photosynthesis and respiration. Plants carry out photosynthesis, and they do this by using carbon dioxide in order to make food, but also plants do carry out respiration using oxygen and producing ATP. So light intensity is crucial in determining whether there is more photosynthesis or more respiration taking place. In high light intensity, like at noon, we're going to see a lot of photosynthesis occurs, or the rate of photosynthesis is going to be greater than the rate of respiration. So more carbon dioxide is going to be used, and that's why we see the carbon dioxide concentration decreases around noon when the light intensity is greater. Also here we see as light intensity increases, there is going to be more carbon dioxide taken up, and as light intensity decreases, there is more carbon dioxide given out, meaning there is more respiration taking place here, and here there is going to be more photosynthesis as the light intensity increases. This leveling off is going to be due to another factor controlling the rate of photosynthesis, meaning light intensity is no longer controlling the rate, but some other factor is causing the rate to level off. So here, as we approach midnight, we see the carbon dioxide concentration in the air is going to increase because there is less light intensity, and therefore less photosynthesis takes place. More respiration is going to be taking place, and that's why the CO2 concentration is going to increase due to the carbon dioxide produced from respiration and less CO2 being used in photosynthesis. Next, we go to effects of sunlight on gas exchange by a leaf. Here we're going to use the hydrogen carbonate indicator to detect the CO2 concentration within the experiment. Remember, sunlight causes photosynthesis to occur, meaning CO2 is going to be used up and the concentration decreases if sunlight is available or if photosynthesis occurs. So we have 4 experiments set up. One is in bright light, and because there is bright light, we expect photosynthesis to occur more, CO2 to be used up, and the concentration of carbon dioxide to decrease. That's why we see a color change of the hydrogen carbonate indicator turning from orange to purple, meaning the concentration of CO2 has decreased. In the dark, there is no sunlight, and that means there is no photosynthesis taking place. The CO2 concentration is going to increase because respiration is going to be taking place. So we see the color of the indicator changing from orange to yellow due to an increase in the concentration of carbon dioxide. And in dim light, we expect less photosynthesis to occur, and therefore the color does not significantly change because photosynthesis and respiration are equal. So overall, there is no significant change in the concentration of CO2, and so the color of the indicator remains. And in the final experiment, this is a control. We place the hydrogen carbonate indicator but no leaf. Since there is no leaf, photosynthesis is not going to occur, and therefore the color of the indicator remains the same. This experiment here is carried out so that we can show that color change is due to presence of a leaf, meaning without a leaf, even when we provide sunlight and every other condition, we see there is no color change, and therefore the leaf is responsible for causing the color of the indicator solution to turn from orange to any other color it has turned. This is kind of the contra experiment. Next, we look at the factors affecting the rate of photosynthesis. When talking about the rate of photosynthesis, we have to consider the carbon dioxide concentration, the light intensity, the temperature, and more. In this graph here, we show experiments carried out to test the specific conditions and how they affect the rate of photosynthesis. I will describe about the processes a little bit later. So I'm going to begin with light intensity. We see as light intensity increases for all concentrations of carbon dioxide and for all temperatures, we can see there is this concentration, that temperature 2020, and then the temperature 30. So for all temperatures and for all carbon dioxide concentration, as light intensity increases, the rate of photosynthesis also increases until another factor limits the rate. And then as temperature increases, I'm going to consider the experiment where temperature changes, but the concentration of carbon dioxide is the same. This is this experiment and that experiment. The concentration of carbon dioxide, or the percentage, is 0.15%, where the temperature changes from 20 to 30. If I draw a vertical line to show the same light intensity, consider this at the same light intensity and the same carbon dioxide concentration. As the temperature changes, there is an increase in the rate of photosynthesis. I'm comparing this result here and that result there. We can see there is a greater increase as temperature changes if we keep all the other factors constant. Then keeping temperature constant, keeping light intensity constant, and increasing the carbon dioxide concentration. Here I'm looking at these two experiments. So temperature is at 20 degrees Celsius, 20 degrees Celsius, and if I draw a vertical line again, same light intensity, I'm considering that result and that result. If we increase the percentage of carbon dioxide from 0.03% to 0.15%, we see the rate of photosynthesis increases, again until some other factor limits the rate. Another factor limits the rate if we see the curve leveling off. In your exams, they'll bring you different experiments trying to study the effect of CO2 concentration, light intensity, temperature, and so on the rate of photosynthesis. To do this, we need to use a water plant because the dependent variable is going to be quantified by counting the bubbles of gas produced or collecting the volume of oxygen produced. So, to test for CO2 concentration, we'll set up the experiment using a water plant. And in this case, since CO2 concentration is going to be varied, we will vary masses of sodium hydrogen carbonate. The sodium hydrogen carbonate is used to provide carbon dioxide to the water plant. And then if you're varying or studying the effect of light intensity, you will set up the experiment using still using a water plant. You're going to position this in a dark room because it's light intensity. And then you're going to use a light positioned at a known distance. You will move the light source closer and farther away from the experimental setup as a way of varying the independent variable, which is the light intensity. And then if you're studying the effect of temperature, still you're going to set up the experiment using a water plant, and you use incubators set to different temperatures as a way of varying the temperature of the water. While doing all these experiments, we have to ensure that the water plant used is the same species and it's the same age, meaning the conditions of the water plant have to be the same. We have to keep the control variables like temperature constant using a water bath, pH using a buffer. If you're not studying the effect of CO2 concentration, maybe if you're studying the effect of light intensity, it means you have to keep the concentration of CO2 constant, and we'll do this by using the same excess mass of sodium hydrogen carbonate. I'm saying excess so that the mass of sodium hydrogen carbonate does not limit the rate. When you're studying the effect of another factor like light intensity, we got to make sure the CO2 concentration is enough so that it doesn't limit the rate, so we have to use it in excess, but for all experiments, it has to be the same. Then if you're studying, let's say, effect of temperature and effect of carbon dioxide concentration, it means the light intensity has to be controlled. We're going to control this by positioning the lamp at the same distance during the experiment. I'm talking about the same distance from the experimental setup. And while doing these experiments, we have to repeat every experiment at each varied independent variable and calculate the mean in order to put the results in a table and interpret them. Next, we'll look at how this experiment could be carried out. You get a beaker, fill it with water. This is a conduit. The outside beaker, or could be a water bath, is to control the temperature. That's why we see the thermometer in here. It's measuring the temperature of the water, and here we see the bubbles of gas coming off during the experiment because oxygen gas is produced during photosynthesis. This ruler is to show the distance of the lamp from the experimental setup, meaning if we are varying light intensity, we'll position it initially here and then maybe move it away and move it away in order to vary the light intensity appropriately. This experiment we've set it up so that the cut area of the stalk faces upwards so that we can be able to see or observe the bubbles of gas as they are produced. If you set it downwards, it's going to be harder because some of the bubbles will dissolve in the water, so it's easier to observe the bubbles if the plant is set up in that manner. We're using sodium hydrogen carbonate to provide CO2, like I already said, and then we use water baths to control the temperature of the water. Light intensity is going to be controlled by moving the lamp at specific distances from the experimental setup, and the bubbles of gas are going to be counted in order to calculate the rate. Next, we go to how plants use glucose. Here we can see glucose from photosynthesis. It could be used for respiration, or glucose could be converted into fructose, and then the fructose reacted with more glucose to produce sucrose, which is a disaccharide. Remember, glucose is a monosaccharide, so sucrose is going to be produced as fructose and glucose come together. So this is going to be transported through the plant in the phloem to be stored. Also, glucose can be converted into starch. Starch is going to be stored. And then we can use glucose to make cellulose. Cellulose is found in the cell walls of plant cells. Glucose can be used to produce amino acids, proteins, and DNA. To do this, we have to add other compounds like nitrates, phosphates, and so on. Phosphates are found in the nucleotides. Nitrates are found in the nucleotides, in amino acids, and so on. So if we want to make DNA and proteins, we need nitrates and phosphates. Then to make chlorophyll, to glucose, we will add magnesium ions from the soil in order to make chlorophyll. And then glucose can directly be used to make lipids, which are the oils in seeds. In some experiments, they use water cultures where soil is not used, but we use a culture solution containing appropriate ions. The key thing is nitrates, phosphates, potassium, magnesium, and so on should be provided because they are necessary for plants to grow. Potassium is useful in enzymes involved in respiration and photosynthesis, so this has to be provided. The nitrates are needed in making amino acids, nucleotides, and many more. Phosphates, like we've already seen, are going to be used in making nucleotides, which include DNA, RNA, ATP, and so on. This soil-free, mineral-balanced culture contains calcium nitrate, magnesium sulfate, potassium nitrate, potassium dihydrogen phosphate, N3-phosphate, and many more. And these minerals all provide the necessary ions for the plant to function properly. During this experiment, aluminum foil is used so that light does not enter into the space here. If light does not enter, then any microorganisms that are within here, if they are photosynthetic, they will not carry out photosynthesis and they will not be able to grow. This includes the algae, which could interfere with the experiment. So if there is no light in there, then they will not be able to grow. But we see the plant, the leaves are outside, so they're going to be exposed to sunlight and photosynthesis will be carried out appropriately. And lastly, we look at mineral deficiency. Here is a healthy bean plant. We can see the leaves are green and it's growing appropriately. But if there is lack of nitrates, this is going to lead to limited growth in plants, and the older leaves are going to turn yellow. We can see the older leaves are the ones down here. They're going to turn yellow because nitrates is required in making DNA, RNA, ATP, chlorophyll, amino acids, and so many more. So if it's not provided appropriately, then the leaves are going to be affected. Phosphate is used in nucleotides, DNA, RNA, and so on. It's also used in making ATP and is part of the cell membrane. So lack of phosphate is going to lead to poor root growth, And the young leaves are going to turn purple. We can see that here. If you compare this to that, you can see this is not healthy. Then we go to potassium. Like we said already, potassium is useful in enzymes involved in respiration and photosynthesis. Lack of potassium is going to cause the leaves to turn yellow and they will have some dead spots like we see there. Also, we know magnesium is used to make chlorophyll, so lack of magnesium is going to lead to insufficient chlorophyll. That's why we see the leaves are going to turn yellow. So this brings us to the end of this video. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.